as you can see quite clearly, that running Xenium prior to IMC didn't seem to have compromised any of our proteins. And we're giving you a couple of examples here, which was really promising. So that gave us the confidence that we could indeed run Xenium first, followed by IMC on the same tissue section. So then, obviously, the million-dollar question was, how does the data look when we compare the two different platforms? So to keep it brief, we looked at various different markers that are associated with immune populations. So at the top row, you can see the data from Xenium. The bottom row, you can actually see the data from IMC. And as you hopefully can appreciate, largely speaking, at the core level, there's a huge concordance between both platforms. So where you would find B cells, for example, from Xenium, you would also find the same populations staining for CD20 with IMC. Same holds true for T cell population. Not just phenotypic markers, you can also delve into proliferative markers like Ki67 or some functional markers. So that globally gave us a bit more confidence that you can actually find immune populations from both these platforms matching really nicely. Then we focus, not everything looks as perfect as that image shows you. We went deep, zoomed in at a single cell level. And that's when we started to see a little bit of discrepancy between the two platforms. So if you look at CD3, you would often find IMC picking up cells that were CD3 protein positive, but we did not see any transcripts that were picked up by Xenium. And you can actually see very nicely in this example here, you can actually find cells that were positive for both platforms, identified by both platforms, but in some instances, you only could see it in one platform, not the other. So that's where the complications started. So if, and we know this really well, biologically, we know not all proteins correlate with their respective RNA. We also know that there could be some technology-based challenges here. So as I said, our ambition was to actually integrate the proteomics with that of transcriptomics, so we could actually really understand what is the molecular pathways that are activated when cells are interacting within the tissue.